Hi everyone, and welcome to our next look at the visual brain. Now we got up to the striate cortex, that is the primary visual cortex, that is area V1. We talked a bit about some of the cell properties in the striate cortex, and I ended up with hypercolumns. Now I want to pick up where I left off and go beyond the striate cortex, so literally extrastriate areas. Now, I did briefly mention area V2. I didn't go into a lot of detail. I want to move on to area V3. If we probe in cells in area V3, we find that these cells are sensitive to moving edges, having a certain orientation. But what does that actually mean? What are these cells actually doing? Well, they're believed to handle perception of forms and also local motion. Local motion as opposed to global motion. Global motion would be if you are looking out the side of a vehicle as the vehicle is driving, everything is moving across your retina. Local motion means mostly everything is stationary, but there's one little thing moving around, potentially. After going to area V3, projections go to the temporal lobe. The next area is, yeah, you guessed it, area V4. And what we know about V4, we've known for quite some time, going back to a case study published in 1888 of a 61-year-old female who had a stroke in this area and suffered what's called cerebral achromatopsia. I have to point out, it is cerebral, meaning it pertains to the brain. You can have achromatopsia that is not due to brain damage. Chroma means color, and the prefix a means not. So the problem that this woman experienced was she couldn't see color. Now, because her stroke didn't affect both hemispheres, she was just unable to see color in the right half of her visual field. Now, otherwise, she could differentiate between things like purple and orange. They just looked different, but she couldn't say, oh, that's the color purple, and that's the color orange. Kind of a bizarre thing. The cells are believed to respond to the perceived color of a surface, not wavelength. I talked about the rods and the cones, and I did mention the wavelengths that they are sensitive to. Rods and cones are sensitive to wavelengths of light. That's not the same thing as color. Wavelength is a physical property described by physics. Color is an internal, subjective, psychological experience. It's pretty remarkable that we have cells in area V4 that respond to perceived color, not to wavelength. I'll get to this in just a second with a visual example for you. I just wanted to point out that after area V4, connections go to the temporal lobe. But let's get back to that perceived color versus wavelength. All right, so take a look at these visual stimuli. And so it looks like there are a bunch of plastic tubes, and then they're joined together at the middle, here on the left and here on the right. But the weird thing is we're looking at these tubes through some semi-transparent gels, an overlay, obviously a yellow overlay on the left, blue overlay on the right. I want you to focus on those X-shaped cross pieces. What color are those pieces? So let's take a look at the one on the left. Okay, blowing that up a bit and bringing that out, kind of bluish. Let's take a look at the one on the right. Blow that up, bring that down. It's pretty yellowish, but oh my gosh, no. They're both exactly the same shade of gray. Yeah, but somehow our mind takes into account that we're looking at these pieces through these yellow and blue things, and we interpret them as having a color. This one on the left looks kind of bluish, and the one over here looks kind of yellowish. But the light that falls on your retina is, well, kind of grayish. There is no color. That's the difference between perceived color versus the actual wavelength. Neat. Okay, I've been mentioning temporal lobe. Let's get to the temporal lobe. Let's see what's happening in there. Not just everywhere, but in specific regions. That is the inferior temporal, or IT cortex. Now, in general, we can actually ascribe what this is doing. These cells are identifying stimuli. And Here's an example of that. Classic study from 1972. They were doing single-cell recording in monkeys, very similar to what Hubel and Wiesel were doing. They had their microelectrode plugged into a single cell in the IT cortex, and they couldn't get it to fire. They tried all kinds of geometric shapes. Nothing was moving. Nothing was firing. I mean, it's just frustrating. This is very painstaking work. And they were going to go to lunch, and one of the researchers was heading out the door and kind of waved on the screen, and his hand cast a shadow, waving goodbye to this cell that they couldn't determine the receptive field for, and the cell started firing like crazy. So needless to say, they didn't go for lunch. They continued to explore. And it turns out that they had plugged into a cell that responded best not to a human hand, but to a silhouette of a monkey's paw. And it's a little bit different from a human hand. The fingers are proportionally longer. That's what this one cell in the IT cortex was looking for. That's what its receptive field was. A monkey's paw. That is very, very specific. Now, further research following on determined other kinds of cells called primary cells. Already, I don't like this naming scheme. Oh, dear. Primary cells respond best to fairly simple stimuli like dots, squares, and ellipses, just geometric shapes. Probing further, do you see where this naming scheme is going? Elaborate cells that respond to more elaborate things, more elaborate shapes, or shapes of a particular color or a particular texture. What's more, there have been face detector cells found in the IT cortex, and this has been found through people with an unfortunate condition called prosopagnosia. It's a bit of a mouthful, and it's Greek, and it means an inability to process faces. More specifically, it's not just due to damage anywhere in the IT cortex, but in the fusiform gyrus. And this is described by Oliver Sacks in his book, The Man Who Mistook His Wife for a Hat. And in that book, Sacks recounts seeing a patient known as Dr. P. At the end of his first meeting with Dr. P reached out his hand and took hold of his wife's head, tried to lift it off to put it on. He had apparently mistaken his wife for a hat. I gotta wonder, what does her head look like? What does this poor woman's head look like that the guy mistakes it for a hat? Okay, it's an unfortunate condition. Okay, let's go to other areas of the temporal lobe, the MT, or medial temporal cortex, also helpfully known as Area V5. I'm sorry, I giggled. I know, some of these areas have multiple names. That's just a fact of life. I'm sorry. Let's take a look at what V5 does. And this is a study from 1983. They discovered a case study of a 43-year-old woman who had a small lesion due to a vascular disorder in her brain, and she experienced cerebral akinetopsia, sometimes called motion agnosia. So look at this word, kinetic is part of this, and the prefix a means not. Akinetopsia is an inability to see motion. She can see the shape of an object, she can tell you how far away it is, she can tell its color. She cannot see motion. It's as if there's a strobe light that goes off every few seconds, and things kind of get updated. Now, she had difficulty pouring herself a cup of tea because she couldn't see the level of tea rising in the teacup. Okay, that's not a major disaster. One day, she wanted to cross the street, and she looked both ways. And she looked down the street, and she saw a car that was parked in the middle of the street. She started to cross the street and was almost run over by the car because it was moving. She couldn't perceive it as moving. That's a bit more dangerous than just some spilled tea. So, Area V5 is responsible for processing motion. And in this unfortunate woman, she actually had it on both sides of her brain. V5 appears on each side of the brain. And in an incredibly rare coincidence, she had a lesion that just knocked out V5 symmetrically on both sides of her brain. So from case studies like this, as unfortunate as they are, they do help us to understand the functioning of the brain in normal people. So we know that the cells in V5 are sensitive to motion and direction of motion of the object. They're not sensitive to color, interestingly. So again, we kind of have a division of labor in the visual system. Some areas handle color, other areas are handling motion. After area V5, it's not the end point. Projections continue on to the parietal lobe. Now, I don't want us to once again lose the forest for the trees. I want to kind of paint you a big picture if I can, but let's just take a look at some of these areas. This is a really nice image. No, I will not have you label any diagrams on an exam in case you think seeing it will kind of help you to understand and remember their names and where they are and how they relate. Here we go. So just a handful of some of the visual areas of the brain, some of which I haven't discussed. So again, getting back to the forest from the trees. So I have talked about these different modules. Some people prefer the name network, V3, V4, V5, for example. Is there a bigger picture? Can we put these separate little modules or networks together? And that's what Ungerleider and Mishkin proposed in 1982, that these different modules or networks are part of two different extrastriate streams in vision. One of them is the ventral pathway, so using some anatomical terms. So ventral means along the bottom of the brain, and temporal, of course, means the temporal lobe. So just to kind of recap how we got to where we are, we started off with parvo cells in the LGN, remember? That went to area V1, then to certain parts of V2, and then to V4, and then to the inferotemporal cortex. What do all of these areas kind of sort of have in common? Recognizing and identifying objects. In other words, what are we looking at? So the common name for this pathway is the what pathway or the what system. Things like color in area V4 are pretty important to knowing what you're looking at. If it's a banana, is it greenish or yellowish, or is it blue? Kind of harder to identify. Things are colored differently. The other pathway is called the dorsal or parietal. So dorsal means over the top, going to the parietal lobe. And again, just to recap, basically a lot of magno connections from the LGN to a certain sub area of V1 to certain parts of V2, then to V3, and from V3 to V4 and to V5 in parallel. And then, as I mentioned, the connections go to the parietal lobe. What do all of these have in common? Yeah, I know V4 is in there. That's just part of this pathway, just to kind of make it a bit more complex and difficult to memorize. What these all have in common is basically locating things. So motion is important. Where is something coming from? Where is it going to? How far away is something? Spatial relationships. And that's why this is called the where system. Knowing what and knowing where something is, surprisingly, according to the brain, those are handled separately. Now, for those of us without any brain damage, with an intact brain, knowing what something is and where it is, that's a unitary property. Those are one and the same. We can't separate them apart unless you get certain forms of brain damage. So here's an image, if this helps you. Also has some cool brain terms like inferior longitudinal fasciculus, which you don't have to know that. So that's the proposal from Ungerleider and Mishkin. It is a theory, but you know what? There is a ton of evidence to back it up. Let's take a look at some of the evidence. Some of the best evidence dated back to 1973 and lesion studies, which means, yes, cutting out or disconnecting certain parts of the brain. And this was performed in monkeys. Yeah, animals are not treated greatly in research and perception, are they? Now, monkeys were given a couple of delayed response tasks. So, the monkeys were kind of hungry, so food is a good motivator. One task is called object discrimination. First, the monkey would be shown an object like a brick or a little cube. Okay. Then, it was presented with a choice task. As you see here, there's the brick and then a cylinder. Now the monkey has to remember what shape it was shown previously, and if they move the correct one, the brick, they will find a little hidden well that held a banana chip. But if you forget and you move the cylinder, you get nothing. You don't get to try again. No second chances. No. The other task was called landmark discrimination. Once again, an object is presented, and then you get this particular choice. The food is hidden in the well that is closest to that object. So if you're keeping track, the previous task was a what task. What shape did I see? The second task is a where task. Where is the object located? Monkeys had to be trained to do this because it takes them a little while to figure it out. And then, oh dear, the task was reversed. Now this makes it pretty difficult. A reversal means if you're shown the brick, the food is now under the cylinder. It's the opposite location. With the landmark task, you're shown the object, the food would be hidden in the other well. That's what the reversal means. So it makes it non-trivial, shall we say. Okay, after learning all this to a particular criterion, getting a certain number in a row correct, that's when the lesioning happened. And Pohl lesioned either the temporal lobe or the parietal lobe. Now those two words should seem familiar. I just discussed those as part of the extrastriate streams. Let's take a look at the effects on the object task first of all. So again, there's the object task, and here's a graph which is a little bit tricky. The x-axis is reversals. So the initial task reversing back to the initial, reversing again, and so on. The y-axis is errors to criterion. Now that means how many errors they made until they could get to the certain criterion of, say, 10 correct in a row. Now let me parse through this. Let's take a look at the two different groups. One group had lesions to their temporal lobe. Pretty clearly, there's a lot of impairment because initially they're making over 100 errors to get back to criterion. It does improve over time, which is pretty interesting. But the main thing is to look at the difference between the temporal lobe lesion group and the parietal lobe lesion group. I know this graph is very low. That just means they're making very few errors. In other words, the parietal lobe lesion group is having no difficulty with this object task. So what is being disrupted? The what pathway. The what pathway in the temporal lobe. Exactly what we would expect. Let's go to the landmark task, which just by its very nature seems to be a more difficult kind of task. So there is a lot more going on here. So they also had a control group, unoperated monkeys, and that is this line here. This is our kind of frame of reference. Okay, this is the best that monkeys can do. Everything else has to be compared to that. Let's go to the temporal lobe lesion group. I know there are some differences between the two curves, but they're showing not a lot of impairment. They're fairly close to the unoperated group. But let's take a look at the parietal lobe lesion group. They make, not as many errors as the temporal lobe lesion group, but importantly, the errors really don't come down, not as much as the other two groups. The parietal lobe lesion monkeys show a lot of impairment. So what is being affected? Their ability to process where, which, as we've seen, implicates the parietal lobe. Okay, well that's monkeys and cutting out chunks of their brain. How about humans? No, we're not going to be cutting chunks of human brains out, but we can do neuroimaging of human beings. Okay, so in this study, people were shown 2 frames. What? Okay, I'll show you an example coming up in a second, so I'll come back to this. There was a brief delay in between. The objects shown in the first frame were moved or replaced with other objects. Now, using the same 2 frames, you can ask 2 different questions, which is really great. You can set it up as an object task, and you're asking people, in frame 2, were there the same objects? Like they might have moved around, but the question is, were they the same objects, yes or no? And you press the button as quickly as possible. You can also set it up as a where spatial task. So whatever the objects were in frame 1 and frame 2, are they all in the same locations? Again, yes or no. Now this isn't super hard. Let me try this with you. Okay, this is going to be an object task. So here's frame 1. Memorize that. Brief delay. Here's frame 2. Are the objects the same? Yes or no? Okay, it's not too hard. The answer is yes. This is not really what we're looking for because while people are doing these tasks, their brain is being monitored, okay? So we're not looking at how well did they do. People can do pretty much perfect on this, but we want to see what parts of the brain are active for the two different kinds of questions that we can ask. Okay, so they're colored in red here, and then blue here and here. And this corresponds to the two different kinds of tasks. The object task involved areas of the temporal lobe, and the spatial task, areas of the parietal lobe. Exactly what we would expect, corresponding to the what versus where pathways. Beautiful. Now I want to further explore these extrastriate pathways, but I want to do that through a particular class of phenomena called visual agnosias. And the word agnosia is coined from the Greek, and it means without knowledge. So gnosis, the silent G, again with the A in the front meaning not. So visual agnosia means without visual knowledge, technically. And what it refers to is some kind of difficulty perceiving, recognizing, processing visually presented objects. And hey, look who made up that word. Now we have seen the word agnosia already in this lecture as part of prosopagnosia. Yeah, so prosopagnosia is a form of visual agnosia. It's very specific. It is a disruption of face perception and processing. People who have this, again, may not be able to identify friends or family members. They have extreme difficulty or maybe even cannot reading facial expressions or emotions. There actually seem to be a couple of types. In one subtype, people don't know that they're looking at a face. You show them a face on a screen, you ask them what they're looking at, and they say, I don't know. They don't even know that they're looking at a face. In another subtype, people know that they're looking at a face because they can count the number of eyes and noses and mouths, but they can't identify whose face it is. In one case, a person with prosopagnosia, they went to a party, even though they were incredibly self-conscious with this problem that they had, they could identify friends if they spoke, but not visually. So here's this person at this party, very, very nervous and kind of sweating, and there's a person across the room that was staring at them. I mean, so they felt very self-conscious, but they kind of gathered their courage and they decided to stare right back. You're going to stare at me? I'm going to stare at you. And the other person didn't look away. They just continued to stare right back. And they felt, so self-conscious. Yeah, okay, I'm a freak. I've got this, brain damage and this face problem. I'm just going to stare right back at you. Until one of his friends came up to him and said, why are you just staring at yourself in a mirror? Now, there are a lot of visual agnosias. People have tried to categorize them even before the term was coined. So I want to go to this and then we'll kind of update it a little bit. Aperceptive is a category of related visual agnosias. The problem is, perception. Yeah, people cannot perceive. That is, they can't form a holistic percept as the rest of us can, the movie that we see in our mind's eye. Now, it could mean that they cannot extract the global structure of an object. They might kind of go, it has a curvy bit to it, which is not the same as saying exactly what it is. Their visual acuity is okay. That is, they can pick up details. They can perceive color. They just can't seem to put everything together. Now, there are tests for this in which people are given shapes or pictures, see if they can recognize them, first of all, discriminate, tell the difference between them, or copy them. And here's an example of a person with aperceptive agnosia in their attempts. So again, you can tell they're not just completely guessing. I mean, the A starts off with 2 slanted lines, and then B is no. X is made up of 2 straight lines, but no, and the O is, you're just, oh man, you were doing so well. So these people essentially are blind. Now let's take a look at what abilities still remain intact. They can reach out and grab objects. You can put something in front of them and say, can you pick it up for me? And they go, pick up what? And you just pick it up. And they will grab it. I mean, they're going to go for it. They're not kind of fumbling around as a person who is completely blind might kind of fumble around until they find it. If you bring them into your lab, would you like a cup of coffee? Make them a cup of coffee. They take a sip. They put it down. Move their mug. They're like blind. They can't see that, right? You move the mug a little bit over to the left. They go to have a sip of coffee and they grab their mug perfectly accurately, even though you moved it. How can they do that? Now, there are some other terms for aperceptive agnosia. I apologize. I apologize that there are all these different terms, but I have done a lot of work to find out that there are these different terms and figured out what exactly they mean. So, aperceptive agnosia is also referred to as visual form agnosia or visual space agnosia. Okay, so now you know, and you won't get confused. Now, we do want to understand the neuropathy of what underlies this. It's typically caused by diffuse lesions. So not a tiny little pinpoint lesion, but lesions to large swaths of the occipital cortex, often due to something like carbon monoxide poisoning, which will affect large areas of the brain, or even things like mercury poisoning. It seems to be a failure to take all the separate aspects of a stimulus, the shape, the color, the motion, and bind them together into a holistic, unitary percept. Again, we believe this is due to damage at a relatively early stage of visual processing. And the second category is called associative. And here, no, the letter A is not a prefix. Associative is what you kind of think it means. It is, okay, you look at a stimulus and you know what it is. It has some meaning to you. Associative agnosia has been called recognition without meaning. These people cannot draw common objects from memory. But if you give them that same test of having them copy shapes or simple pictures, they can do it. It's kind of painstaking because it takes them a long time, like 20 minutes. And then they show you their results and you go, okay, what did you just copy? And they go, I don't know. And here's an example of some of that. So I think they're actually pretty good. Like, I don't know if I could copy that pig as well as this person could. So again, they've spent, 10 minutes copying this painstakingly. You ask them, okay, what is that? And they'll go, I don't know. Now, initially, it was suspected that these people just cannot name objects. They can't put a name to it. So you show them something and you go, what is this? And they say, it's round, so it could be just a language problem. But if you allow them to use their other senses, this can help them to identify the stimulus. A subtype of associative agnosia, so one specific subtype, is called visual object agnosia. Don't mix this up with visual form agnosia, okay? In visual form agnosia, there was one person who had this and described an object as, quote, a convoluted red form with a linear green attachment. So clearly, they can kind of tell shape, they can tell color. What are you looking at? I don't know. Well, why don't you smell it? And the patient smelled it. Oh, it's a rose. And this example also comes from Oliver Sacks. The neuropathy is not consistent. That is, we don't get a specific pattern of brain damage that's giving rise to this. It's probably because there are many very different subtypes of associative agnosia, actually. And it's probably that this category is just too broad for us to get kind of a consistent picture. We can generally say that there seems to be a disconnect between visual processing and then deeper knowledge, so semantic knowledge. So what it looks like, and then connecting that to, oh, that's what it is. Which, again, if you don't have brain damage, that seems to be one singular process, but this gives us evidence that they are two separate things. So the thinking is now that these two categories of apperceptive and associative, they're too broad, and we should just go to the specific types, like visual object agnosia, and let's not try to put them into categories. So here's some specific agnosias by Martha Farah, who literally wrote the book called Visual Agnosia. I'll just point out some of these. There's one called category-specific agnosia, in which people, based on visual stimuli, cannot identify living objects, or perhaps the opposite, non-living objects, or metals, or fruit, or vegetables, or all kinds of very, very specific things. All of these are based on individual case studies. Orientation agnosia means that people can recognize objects that are rotated, whereas those of us without this kind of agnosia would have difficulty. If you give them the picture copying test, they might actually copy a drawing perpendicular to the original. So the original might be the Eiffel Tower, and they'll copy it, but it's sideways, and they won't be able to tell that they copied it at a 90-degree angle to the original. This next one is a little bit more complex, called simultanagnosia. Kind of looks like the word simultaneous. That might be important in understanding this. So my first definition here is kind of general. Inability to perceive more than one aspect and integrate into a coherent whole. So there seem to be two subtypes of this. One is called dorsal simultanagnosia. And what the hallmark of this is that if you show people this kind of a picture, it'll take them a while, but eventually after kind of looking at it and staring at it, they'll say, I see a hammer. And you go, very good. What else do you see? And they'll go, what? It's just a hammer. Bring them the picture the next day. Show it to them, the same kind of a thing. They'll spend lots of time looking at it. I see, it looks like a bottle or a milk jug. And you go, very good. Do you still see the hammer? And they'll go, what? They can only see one thing at a time. They may not be able to tell you where things are, interestingly. This is typically caused by bilateral damage, so both hemispheres in occipito-parietal regions, which is not terribly specific. The other subtype is called ventral simultanagnosia. Now, people here can perceive more than one at a time, but they cannot identify more than one at a time. They might be able to describe one part of a picture, but they can't tell you the whole story. So this scene is part of a standard set that is given to people to test them, and they might, look at this for quite a while and then say, it's a picture, I think it's a highway, an old-timey car. Okay, you're not wrong. Anything else? Nope. Show it to them the next day. They'll tell you there's a forest. Show them again. Oh, it's a man riding a bicycle. They can't put it together and tell you the story of the man whose bicycle broke down and now he's trying to hitchhike and get a ride. People with this subtype can localize objects. They can tell you where the things are, the cars on the right, for example. Damage is in the left inferior temporo-occipital cortex. Sounds awfully specific, but you know what, not really. Some kinds of visual agnosias affect our ability to read, like pure alexia. So again, the prefix a means not. People here cannot read words, at least not normally. What they have to do is read letter by letter. It takes them a while to figure out the first letter, R. That's like a O or it's an E, and the next one, A. And they have to remember the letters and read in a very painstaking fashion. Now, if they suffer this kind of brain damage later in life, when they can read and write, they can write to dictation. They can take a letter. But then if you ask them to read it back, read what they've just written, they kind of have to go letter by letter by letter. Interestingly, this has some similarity with ventral simultanagnosia. So people have proposed there might be some overlap here. The last one I want to talk about is called topographic agnosia, in which people do not recognize scenes or landmarks, And if you navigate using landmarks, you're going to get lost pretty easily. Okay, I think that thing is happening again. We're losing the forest for the trees, going into all these specific agnosias. What is the big picture? What can these tell us? Okay, the reason why I took this little detour is because I wanted to lay some groundwork for the double dissociation of the extrastriate pathways. Now, the gold standard in neuroscience, if you're proposing that there are two independent pathways, is you would look for evidence. Evidence that one pathway is impaired, the other is intact, and vice versa. So maybe the case studies of these people with these different specific agnosias can actually provide us with this double dissociation that we're looking at. That means sifting through the case studies, testing people. You know what? Brain damage is not cooperative. It's difficult to find cases that are perfect exemplars of one particular agnosia. Again, the brain damage might have affected multiple parts of the brain, and they might have multiple agnosias or different degrees of impairment. It's not kind of simple in black and white. But let's see if we have the components that we need for a double dissociation. Let me go to another kind of agnosia called Balance syndrome, named after its discoverer, Rudolf Balint, in 1909. In this case, the patient that he reported on had bilateral damage to the superior posterior parietal lobes. And this kind of brain damage does not happen every day. It is incredibly rare. And this person had a number of problems, including optic ataxia, which means if there was an object that is reachable and in their field of view, they wouldn't be able to grasp it. They're kind of fumbling around until their hand accidentally hits it. They also have optic apraxia. That is, it's really difficult for them to guide their eye movements or to move their eye from staring at one thing to looking at something else. Also have simultanagnosia. Now, what they can do is recognize a single object, but they can't tell you where the object is or point to it. That's pretty interesting. What this looks like is an intact what system, but a damaged where system. So 1/2 of what we're looking for, this is a single dissociation. What we're now hoping to find is the mirror image of Balint syndrome. And let's go to this study by Milner and Goodale, some Canadian content. They studied, case study, an individual known by her initials in the literature as DF. We know her name is Diane. We also know that Milner and Goodale refer to her as D. She experienced carbon monoxide poisoning, giving her occipitotemporal brain damage. So again, that kind of widespread damage to her brain. As a result, D has visual form agnosia. She can't discriminate objects, tell you their shape, their orientation. She's been tested extensively. She can't recognize or draw an apple or a book by looking at simple line drawings of them. So this is her attempt at copying. No, not even close. Now, she can draw them from memory and do a fairly good job, or just by holding the object in her hand. What can she do? She can perceive colors and textures. So, for example, she can tell that she's looking at a banana because it has a certain yellow color and a certain texture to its surface. She surprises herself by being able to reach out and grasp objects, so she could reach out and grasp the banana. Now, Milner and Goodale came up with some lab-based tests. She performed two similar, but importantly different tasks. One task is called orientation matching, in which you're given this kind of card, and you're asked to hold the card at the same slant as the slot. Okay, so not putting it in the slot, just holding it up at the same slant. On the left, we have D. On the right, we have a control. The control is basically perfect. They changed this task just slightly, but importantly, it's called visuomotor posting. And now, same card, same slot, the task is put the card into the slot. And now, Diane is about as good as a control person. Looks like her what system is pretty severely damaged, but her where system is intact. Now, some people have a bit of an issue with the term where. It seems a very passive, oh, I know where that is. So some people have proposed that instead of calling it where, we should call it the how pathway. This is really its role and its function. It helps us to interact with the world. We know how to place the card in the slot. It is visually guided action. One last thing to talk about, which doesn't fit into the category of agnosia, but it's still important to look at, called, paradoxically, blindsight, first described by Joseph Weiskrantz. Weiskrantz published on this. It was difficult for him to get these studies published because people didn't believe that what he was reporting was an actual thing. But no, it was an actual thing. And in this case study from 1974, reported on a patient known by their initials DB, who had their right striate cortex, that is area V1, removed. They had some abnormal blood vessels. This person was at a high risk for a stroke, so they preemptively removed the right striate cortex only. As a result, DB could not detect stimuli if they fell on the right half of either retina. That information does end up going back to the right visual cortex. For the other half retinas, vision was 20/20, like no problems. So even though DB could not detect stimuli or identify them, he could point to spots of light. Wait, what? So if you showed a point of light, DB could point to it. And you go, well, what are you pointing at? And DB would say, I don't know. I have this feeling like I should point here. Well, do you see anything? No. Couldn't see a thing. He could guess whether a stick or a line was horizontal or vertical, but did not have any subjective experience. And you could see how frustrating this is. Here's a quote from him. I couldn't see anything. I couldn't see a darn thing. Such language. Some people who have blindsight can identify facial expressions correctly 90% of the time, even though they think they're looking at nothing. This is amazing. I have a video that I'm going to insert here of a person who does have blindsight. They're taking this patient from one room to another, and they didn't tell the person that there would be obstacles. And just watch the video and see what the person does. Okay, so pretty obviously that patient was picking his way very carefully around those obstacles. When they reached the second room, they asked him about the obstacles, and he went, "What obstacles?" He had no knowledge that he was carefully picking his way around these boxes and things. Apologies for the poor quality of that video. Blindsight is associated with damage to area V1. This is what we know about it. What this tells us is that there must be some pathway that goes from the eyes and it goes to extrastriate areas, but it must bypass area V1. So there's a couple of possibilities that haven't been identified. It might be the tectopulvinar pathway, or maybe some pathway that goes from the LGN to area V5. Yeah, it's possible that there is a pathway that hasn't been identified yet, which is, I know, kind of bananas. Okay, what does this tell us? Well, this tells us that vision, which is something that we think of as a conscious experience, isn't necessarily. We do have this geniculostriate pathway that we rely on for conscious vision, but we're also getting information from our eyes that is processed unconsciously. Pretty neat. I wanted to give you a quick summary of where we started off with. We started off at the eyes with the rods and the cones, bipolar cells and ganglion cells. And here's a bit of a summary. So there is a lot of feed-forward connections, V1 to the parietal lobe, and then we've got these dashed lines as well. There is feedback that comes back to V1. Not studied in as much depth. We don't know as much about these as we do the feed-forward pathways. Although I've presented these as two separate pathways, there does seem to be some interaction between them as well. So it's not as simplified as we might at first assume. I want to summarize just a little bit more. This is quite a bit out of date, but this is the most recent one I have. This is what we know about the visual system dating to 1991. Okay. This is what's known, and since 1991 we've only added to our knowledge tremendously. So let me explain what we're looking at here. RGC is retinal ganglion cells, M and P, the magno and parvo layers of the LGN, and then all the feed-forward connections. Now if you're like me, you start at the bottom and you work your way up to the top, which is where vision must take place. ER is the entorhinal cortex, which surrounds the HC, or hippocampus, which is not the endpoint. From the hippocampus, connections get sent out to the cortex. There is no endpoint. So where does vision take place? Everywhere. Like everywhere in here. All of this is important. If you wipe this out, well, you're going to be completely blind. If you wipe out area MT right here, you're not going to perceive motion. If you knock out area V4, there goes your color vision. These are all aspects of our visual experience. We need them all to work together to create the holistic experience of seeing. But how exactly does that work? That's something that we're going to come to in the next lecture topic. But for now, check out the E-Class lecture quiz.